Hello, my name is Simran and I'm going to discuss about the microbiome diversity analysis in this video. So basically, first we need to know what is microbiome diversity analysis. Microbiome diversity analysis is the measure of the distribution of community of the microbes depending on the different types and number of the species. Also, microbiome diversity, it's measured by the richness and diversity of the microbes. So basically, richness is like how many, what's the number of species that are, what is the number of a particular species of bacteria that are present in that particular area. I'm just giving an example of bacteria, it can be any microbe. And diversity is how many kinds of species are present in that particular area. So next is we need to see what is the value of the richness and diversity. Higher the value of the richness and diversity, the more stable and stronger is the immune system. So that's the major application. In this case, we are going to examine different items and check the presence of the bacteria, whether they're present or not. So this is our experimental setup that consists of the water bottle, shoe, wallet, phone and glasses. So these items were collected by different persons. In this experiment, there were 5 persons, including both males and females, and with different ethnicity, and they were examined. So these are the sample details. The first column gives the sample number followed by the item type, whether it's a water bottle, whether it's a shoe, phone or glasses, and then who collected that particular sample, and we will check whether that is a male or female and what's the ethnicity of that particular person. Here we are going to use mainly we'll check the item type only. We are going to focus on the item type only. So in a statistical analysis, basically we need to form an hypothesis, the null hypothesis and the alternate hypothesis. It's a statement in which we say what result we need, and we need to check, we assume the things. Here in this case, we are assuming the null hypothesis that there is enough evidence to show that the bacteria is present on the different types of objects, and the alternate hypothesis is that there is no evidence of the presence of the bacteria on the different types of objects. So basically, for analyzing this data, we are using the Chime. It's a software that is we can use in the Jupyter notebooks with the Python. It's basically called the Quantitative Insights into Microbial Ecology. It is an open source pipeline for performing the microbial analysis. So Chime is like we can input the raw sequence data into it to get the desired output. So the raw data that is mostly generated by the Illumina or some other platforms like Publication or Statistics. So there are 2 types of files. I'm discussing about the 2 of them. The one is the QZA and the other one is QZV. QZA is called the artifact files and the QZV is called the visualization file. So for the semantic types, the type and format of the specific data types, example, say first Q table or a metric, it depends what kind of data you are using it, and the visualizations, the output data can be viewed with this link, that's a Chime view. So you can just drag and drop your file over there to see what is the analysis. So working like this, we have got the analysis. The first thing is feature details. The first one is a table summary. So we have got the number of samples to be 20, number of features to be 1144, and the total frequency is 11,416. We can see that the frequency per sample is like the minimum one is 0 and the maximum one is 1392. And moving on further, the first quartile is 239.75. It means that 25% of the population is below this number. So in the same way, there is a median frequency of 536, which is nearly quite less than half of the total of the maximum frequency. So like we can see here, the interactive sample detail, that is without sampling. Here we have got the ethnicity. On the basis of the ethnicity, we can see that Australia has got the maximum number of samples, whereas when we check the iron type, the water bottles have got the maximum number of samples, and most of the samples were collected by males, and the maximum number of samples were collected by Michael. So moving on further, when say I put the sampling value to be 314, then the maximum item type that remains the water bottle. But as we move on to the person, Jonas has got the maximum number of samples. Further, the males, they are again leading the females, but in the case for ethnicity, South East Asia and Australia have got nearly the same number of samples. So the first thing we need to know is what we are going to do. So what are the steps we are going to follow for this analysis? So the first thing is pre-processing of data, which includes the error correction. So we'll be discussing this in detail in the later slides. Then the second one is picking up features and representative sequence. So first thing is assigning the taxonomy, is basically classifying which kind of bacteria are we having, align the sequence, and infer phylogeny. So we are checking, okay, we are getting the information about the phylogeny, and then calculate the alpha and beta diversities. So before going on to anything, we need to demultiplex the data, because we have got the multiplexed files. So firstly, we need to know what is the demultiplexing. It's the breaking up of the data into individual sequences. Basically, it consists of the mapping file and the sequences. Here, after the demultiplexing, we have got two sequenced data. The first one is the reverse, and the second one is the forward sequence. So that's the summary of the sequence. The minimum one is 1013, and the medium one is like we have got in millions of the sequences. That's all based on the counts, like how many, what's the number for the different kinds of sequences. Here, as we discussed, we need to go for the error correction, that is the denoising the data. This will be done by the data to pipeline that was used to truncate the sequence and reduce the error, which was checked by the visualization of the QC results. Basically, we are going to measure the truncation level by checking the Phred scale, that is like checking up the Phred score. So if the value of the Phred scale ranges from nearly 10, the accuracy is nearly 90%. But if the value increases between 20 and 30, the accuracy will become 99.9%. That's a very good one. Secondly, we have got the interactive quality plot, and we need to reduce the error on the basis of that. So we have got the forward reads and the reverse reads in these two graphs. So these plots were generated using a random sampling of 10,000 out of those millions of sequences without replacement. Talking about the forward reads, we can see that the blackish area shows the noise in the data. So we will get, if we want to truncate the data, will get the maximum number of sequences starting from the sequence space to be 60 and going on to 180 or 200, it depends. And the other one, for the reverse reads, we can start from 60 or 80 and go on till 160 or 180, so that we don't get the unwanted things, unwanted sequences, so that we get the more accurate results. Here, the next step is the taxonomy, assigning the taxonomy. So that's the provenance graph for the taxonomy. Here, when the things were classified, when the features were classified, we have got two kinds of bacteria. One is the K bacteria and other one is unassigned. So talking about the item types, we see that the K bacteria has got the maximum number, maximum distribution, and the unassigned is quite less as compared to the K bacteria. Now talking about the persons, again the K bacteria is leading the unassigned bacteria. In the next one, we are talking about the males and females on the basis of the gender. Even this is the distribution, how the bacteria were distributed on the samples taken by the males and females. Further, we need to work on the diversity matrix. This can be seen by the PCOA, that is the principal coordinate analysis. The other one is the scree plot, which shows that the points on the axis plot are more principal as compared to the on the axis 2, and then it is followed by axis 3. So the other one is the provenance graph for the phylogeny. It's like the after the demultiplexing of the sequences were aligned to form the tree and for the phylogeny. Further, the main thing we are going to check is the alpha diversity and the beta diversity. So alpha diversity, it is the variation of composition of species within the habitats of the species, while beta diversity is the variation of the composition of the species between two habitats. So basically alpha diversity, we are checking the variation within the habitat, but in the case of beta diversity, we are checking the variation with the different habitats. Here we have got the provenance graph for the alpha diversity. It's like first we need to demultiplex the sequences, and when we get the output sequence, these are aligned and they are assigned the taxonomy fed into the tree, and then the phylogeny is inferred. From after working on these steps, the next step is the finding out the alpha diversity. Here are the box plots for the alpha diversity. So for the classes, there is only one kind of bacteria present, and the phone one kind of bacteria, and in the case of violet, there are two kinds of bacteria present in the sequence. So that's the alpha rarefaction curve. The first one is sequencing depth versus OTUS, and the other one is the sequencing depth versus the number of samples. So basically what we have got for considering all the groups, the p-value is 0.25924. So we need the p-value to be less than 0.05 to accept the null hypothesis, but here in this case for all the groups the p-value is coming to be 0.25, which is quite higher than the 0.05, so we cannot accept the null hypothesis. So we'll go with the alternate hypothesis, which is we haven't got any enough evidence to show the presence of the bacteria on the different items, but here in the case of glasses and phone, the p-value again is quite high as compared to the 0.05, that which shows the significance level. So it's again the value is high, so we cannot accept the null hypothesis. So again we haven't got the enough evidence to show the presence of any kind of bacteria in the item types that are present, but so we need to check whether we have done something wrong in the analysis, or we can verify that by taking the other samples as well and checking the environment. Moreover, it depends how we have taken the sample. So for the best results we need to simulate our experimental setups and then check again the results. So this was all about the microbiome diversity analysis. So thank you.